Thank you very much for that introduction, Julia. And the title of my talk, as taken from Sarah, is Translating the Human Cell Atlas. But unlike Sarah, I will be mostly focusing on the human lung today because that's what I've really been working on over the last 7 years or so. So we've been using single-cell data and developing analysis tools to really ask the question of what cell types and states are there in the human lung, and how do those cells interact and talk to each other to build cellular niches. And all of these questions are relevant to the human lung in health, but today I'd really like to focus on how we can use these approaches in terms of also developing drug targets, so for drug target discovery. Now, most of what I'll be talking to you, at least in the early part of my talk, will be on some discoveries in the healthy human lung, but I will come more to the drug discovery at the end. So for those who are not so familiar with the Human Cell Atlas, first a little bit of an introduction. So the mission of the Human Cell Atlas is to create a comprehensive reference map of all human cells, both in health, but also very much with a view to treating disease. And in order to do this, our go-to tool is really single-cell RNA sequencing. So we do this by taking a tissue, dissociating it into single cells, and then, of course, sequencing either in a plate-based or fluidics-based systems, and then we get the single-cell data out. And of course, this whole analysis part here has been developing hugely as well, using a lot of machine learning and AI tools that now really help us to interpret this data and identify single cell types from that data. And this has been applied to the lung by many groups over the last decade or so, and here I've highlighted a couple of already published studies that have defined the cell types in the human lung. Here's an older study from our lab where we generated an asthma cell atlas, and then another quite highly cited paper from the Krasnow lab, but there are many, many more papers. But importantly, many of the groups who've generated this kind of single-cell data have actually come together. And then last year, the HCA Biological Network published an integrated cell atlas of the lung in health and disease. But even though the title of that paper says it's an atlas, that's actually cheating a little bit because it was primarily a list of cell types, because what was very much missing from this atlas was the spatial component. How do those cell types that we've defined really sit in space in the tissue? And of course, that's why the second tool for the Human Cell Atlas is really the analysis of spatial gene expression. And for that, we've used a number of different tools, things like Visium, single molecule FISH, IBEX protein staining, and also in situ sequencing, although I won't be talking about that today. So the study I'll be presenting today really wants to use these tools in order to move from a suspension atlas to a spatial atlas of the lung. So the way we set up our study is to sample macroscopically in a number of different locations, trachea, different size bronchi, and then upper and lower parenchyma, and then carry out single cell and single nuclei sequencing, but then complement that with also microscopic analysis where we're doing spatial transcriptomics with Visium. And this work was actually published last year, and the two postdocs really driving the analysis and the experiments were Elo Madhusoon and Amanda Oliver. So this experimental setup gave us a data set of about 100,000 transcriptomes, which on this slide just shows you in very broad cell type annotations. But Elo was able to really dig into the details, and in the end, we defined as many as 80 different cell types when previously there were only 58, so really adding to the granularity in which we understand the different cell types. I'd also like to just briefly point out that it's very, very helpful to have different modalities. So, for example, single cell and single nuclei. And a particular example here is, for example, the chondrocyte, which only came from the single nuclei data because, as you can imagine, it sits inside the bone, and it's just really, really hard to get at. So when you do a physical dissociation, which you can do for the nuclei, you capture those cells. Also, you can see here the usefulness of having different regions of the lung, because of course the chondrocytes primarily come from the upper airways rather than the parenchyma. So whilst I'm giving here just the broad overview, I'd now like to come to the spatial data, and this first slide just really gives you a proof of principle of how we do this. So up in the corner here, you see a tissue, a cross-section of a small airway with the lumen and the epithelium and then the tissue around it. So this section gets overlaid on a glass slide where there are multiple voxels with different local oligos. Sorry, I turned away there, you couldn't hear me, apologies. So you then take the tissue, overlay it on this glass slide, the mRNA diffuses out and gets captured by the oligos, and then you make a library, sequence it, and because each of those oligos had a barcode, you can then map the transcripts back into space. And just as a proof of principle that this works, we've shown at the bottom the mapping of single-cell data back into that space, and you can see that around the lumen we have the ciliated cells as expected, whereas in this region here, where we have a little bit of parenchyma, we can see alveolar type 1 and type 2 cells in the lung. So that's just how it works in principle. And now I'm showing you some new data where Elo has taken all the fibroblasts and allocated novel cell states. And I just like to, so all the red ones are new ones that you managed to annotate, and I just like to focus in on one here, the one in blue, the peribronchial fibroblasts. Now, normally when you analyze your single cell data, if you have a cluster, you say, great, it's a new cell type. But of course, it's often very difficult to understand whether that's really a new cell type or whether it's just an artifact of your computation analysis. But if you now have spatial data, you can ask whether this new supposed cell type looks different. And that is really very nicely illustrated here because the peribronchial fibroblast maps around the bronchi, which is why we named it that way, whereas the adventitial fibroblasts map throughout the tissue. So you get very nice orthogonal data that this is really functionally different. And the computational tool that we use to do this mapping is called cell to location, which was developed by Vitaly Kleshtevnikov at the Sanger. And so we've gone on to use this Visium data to identify a gland-associated niche in the airways. And this came about when we first annotated plasma cells, both IgA and IgG plasma cells, and then mapped them onto the Visium. And as you can see in the top two panels, the IgA really mapped to a region of the glands. And then there's a sort of a larger section at the bottom where you can see that all the submucosal gland cells, like the serous cells, completely overlap with the IgA plasma cells. And this was not just true for one slide, but when we looked across multiple different Visium sections, we saw that the submucosal gland cells all map together with the IgG, whereas the IgG primarily maps to the immune infiltrate. And even though I'm a huge aficionado on single-cell RNA-seq, we did want to test that this is also true at the protein level. So we did some protein staining here, a collaboration with Nathan Ricos from Mena Cladworthy's lab. And there you can see that the EpCAM stains the glands. And then in the middle panel underneath in blue, you can see that IgA maps to this region, whereas there's very little IgG there. Incidentally, we also see a lot of T cells in this region, which I'll come back to later. So I've now shown you how we can use the single cell data, use algorithms like cell to locations, and map that data back into the tissue context so that we really begin to understand which cells sit next to each other. And then we can use another algorithm, a database that was built in our lab, CellPhoneDB, to understand how these cells really talk to each other. So CellPhoneDB is effectively a database that has curated knowledge of which ligands bind to which receptors, and often, importantly, it also includes heteromeric receptors. And you can then use this knowledge to predict from your single-cell transcriptional data which cells interact with which other cells. And so Amanda did this for the submucosal gland cells, and here you can see that particular what stood out is that the serous and mucous cells produced a molecule called CCL28 that interacted with CCR10 in the B plasma cell. And indeed, it was only the gland that expressed the highest levels of the CCL28. And conversely, for the ligand, we actually saw that IgG, which that the receptor CCR10 was only expressed relatively lowly in IgG, but much more highly in IgA, which probably accounts for some of the specificity that we saw. We then went on to do lots of validation, which is in the published paper, so I won't take you through that, but I'll show you some summary. So I've told you that we have the gland cell types that produce CCL28 and other molecules. They, in turn, recruit IgA plasma cells via CCR10. There are other molecules such as IL-6 and APRIL, which are produced by the gland that again interact with plasma cells and their receptors to increase the longevity of these molecules and also increase the Ig production. I've mentioned that there are T cells in this region. We know that they get recruited via MHC interactions and then in turn give help to the B cells. We then get increased IgA production that goes into the lumen where it can bind virus. So we can, so what I'm really trying to highlight here is that we can, from the single cell data, really start to begin to put together the cellular circuitry, how these niches are established, how is the cell first attracted into this area, how is it kept there, and then how does it perform its function in terms, in this case, of producing IgA. And of course, this is also clinically relevant because many of these immune modulators that are on this slide have already been used by pharma to produce monoclonal antibodies that are being used in various immune indications. I leave the gland there and move on to the vasculature, which Elo also annotated in a very detailed way. And in particular, because of the lung, we have the pulmonary circulation and the systemic circulation. And she was actually able to distinguish those on the single cell data. And again, we validated this with the Visium data. At the same time, Anna Maria Kushba and Ling Yu in the lab collected a whole lot of single-cell data that had been generated by other laboratories as well as our own data, and actually started to generate an integrated human vasculature cell atlas. And as expected, within this vasculature atlas, we see both venous and arterial as well as capillary cells, and we find cells that have common endothelial markers as well as unique markers for these different cell types. But importantly, whilst we can look for commonalities between these cell types, we can now also start to see what are the differences between the same endothelial cells in different tissues. And what we find particularly is that capillary cell types are the most organ adapted. And so this heat map here actually shows you a sort of dissimilarity plot that shows the uniqueness of capillary cells in the different organs. And of course, when you have this dissimilarity, you can now imagine that this can lead to different drug responses. And this is where we've developed another tool or computational tool in the lab that we call drug to cell. And this was published by Kazumasa Kanemaru and James Cranley in the lab last year in Nature in a heart cell atlas. And what they did is they take the Chembl database of drug target pairs, so there's about 20 million or so, and then link this data with the single cell and single nuclei data that we have in the human cell atlas. So you can now, for any drug that you apply, you can predict which cell type will interact. And as this talk is on the lung, I'm giving just one little example here where we've looked at different drugs, and these happen to be antidepressants, and then ask where in the vasculature do these cells actually, these drugs actually interact. And it turns out that these two drugs interact with the lung microvasculature. And actually this was a very good finding in a way, because it was known that the use of these drugs can cause a risk of interstitial lung disease. So by applying this drug-to-cell algorithm, we were really able to give a molecular explanation for this off-target effect. And now, while in this case I'm just giving an example of an off-target effect, you can of course also think about using this type of approach to do drug repurposing and drug life cycle management. And of course, the previous speaker talked a lot about having the right drug for a target, but you also have to be really, really sure that you know whether that target is expressed in the right cell type. And this is really what this kind of approach with the human cell atlas data can give you. So with that, I've given an overview of the types of data we generate, but as part of that, we've now really believe that we've also sort of have developed a strategy for target discovery, because we have algorithms that allow single cell data to be mapped into the spatial locations, and then using things like cell phone DB and drug to cell, we can understand ligand receptor interactions and potentially how these can be targeted. And while all of this has really been in the healthy lung, we can of course apply this in the disease setting. So in disease, we would like to understand cell type and disease-specific gene expression in order to find molecules. And I think it was Steve who earlier talked about the balance and imbalance of, for example, gene expression across cell types. And of course, really, that is what the cell atlas is meant to find out. By having the healthy reference, we can now really see what is perturbed in disease. If we combine this also with analysis of spatial gene expression, we can understand which targets are spatially relevant, and then hopefully also druggable or accessible. So if we want to invest time and effort into this kind of drug discovery using single cell, what's the actual evidence that it makes a difference? And so in order to look at this, Emma Dan in the lab, a PhD student who's only just finished, really dug into that question. And she took inspiration by the studies by Nelson et al. That wanted to ask whether genome-wide associations actually help in drug discovery. And of course, there was the very famous paper he published in Nature Genetics that said, yes, if you have a clinical target with genetic evidence, then it is more likely to reach a phase 3 clinical trial. So Emma tried to recapitulate this, but rather than using genetic data, she then now wanted to ask about single-cell omics data. So she defined two different types of single-cell data, either cell type-specific expression of a target, where a gene is expressed in a cell type-specific way in disease-relevant tissue, or disease cell-specific expression, where there's a disease tissue with a pathological cell type that expresses your target. And when she then took gene-drug pairs in trial from the Open Targets database and asked if any of this data is available, is your likelihood of success improved? And the answer is yes. It was about threefold improved for phase 1, phase 2, and phase 3 clinical trials. And what's also very nice to know is that single-cell omics provides a greater pool of targets than genetic data. So here is just a graph of clinical targets which are greater than phase 1 for the 30 diseases that she had picked out to study. And what you can see here is that in red at the top, we have the targets with genetic support. In this case, for phase 2, there were 18. And compare that to the number of genes for which there's single-cell omics support, which were over 300. So you have a much bigger space in which to search. And importantly, the overlap between genetic evidence and single evidence is actually quite small. That's shown what's in the purple bars at the top. And so really these two technologies are complementary and therefore will hopefully really help. And lastly, a really nice thing is that you can get single-cell evidence from really quite a small number of people. The lung study I showed you was carried out with 7 patients, whereas, of course, in order to do a GWAS, you probably need tens of thousands of people who all need to be genotyped, and is a much, much greater investment. So with this belief that we can use single-cell data, we would really like to apply the data and the tools from the Human Cell Atlas to clinical samples. And in particular, we believe that if we apply single-cell and spatial technology, we can, first of all, define disease-specific expression, which, as I've just shown you, is, of course, relevant for drug target success and, of course, for target identification. But also by really profiling the patient cohort prior to treatment, hopefully we'll be able to really get a really detailed, fine-grained molecular footprint of disease, really then allowing us to define new endotype and move precision medicine, not just for cancer, but then for many other common diseases, like for asthma or COPD and others. And then the other area in which we believe we can really apply this is organoids and model systems. So in the lab, we've not just studied the adult lung, but there was recently published a paper on the developing lung in the first and second trimester of development. And that, of course, gives us a blueprint for how the lung develops normally in pregnancy. And now we can compare that to how lungs or allele cultures develop in vitro, and then we can start to use single-cell sequencing and really pinpoint the molecular changes that may not be happening to the same extent in vitro, and then hopefully adjust the model so that we get a much more lifelike model system that will allow us to then do experiments in vitro in the lab that are more meaningful maybe than the mice that we can't cure of cancer. Or that we can cure of cancer. And then lastly, of course, organoid models will be incredibly important to really understand disease mechanisms. But again, by following that with single-cell sequencing, we feel we have a much better handle on really starting to understand disease mechanisms. So with that, I'd like to finish and just summarize. I've showed you the data that we've generated for the lung and the airways. We've defined cell types and cellular niches, and in doing so, we've really set up a computational pipeline that helps us to identify these cellular niches and how they work in health and disease, and hopefully that's translatable. I've told you about the IgA immune niche, how we can start to understand cell-cell interactions and regulatory circuits, and again, how that could potentially be turned into drug targets. And I've talked to you about the drug-to-cell algorithm and how single-cell omics can be used to help expand the universe of drug targets. And so with that, I'd just like to acknowledge all the members of the lab. In particular, the lung work was driven by Elo Madusun and Amanda Oliver. Anna Maria Kushba and Ling were the leads for the vascular atlas, Kasumasa for drug-to-cell, and Emma Dunn on the open targets that I ended with. And of course, I'd like to thank all of our funders and importantly, the organ donors and their next of kin who have allowed us to use samples. And with that, thank you very much for your attention, and I'd be happy to take questions.
Thank you, Kerstin. That was incredible tour de force. Right, questions from the audience. I can see a hand over there. Yes. I don't know where my microphone's. Shout. Or microphone's coming, sorry. No questions.
Hi, wonderful talk. I think I liked your graph the best, in which you said the single-cell data can expand the drug space in comparison to just the genetic. So what is it about the single-cell data that expands this space?
So we're actually digging into the data deeper. At the moment, the data that I've shown you has always been the sort of protein universe against those that are successful, against phase 1, phase 2, phase 3. We're now digging in a little bit deeper and also asking, is it actually from the universe to phase 1, which would be primarily safety data. So maybe by really knowing that something is quite tissue specific, you just have fewer toxic side effects. We're also looking at whether there is a progression from phase 2 to phase 3, but that we haven't quite got the data yet because we needed to expand the data set to get significant results. So I'm afraid I can't give you the full answer to that yet, but we certainly feel that it's got quite a big role in predicting things that might be toxic, or putting the other way around, that just if you happen to choose a target that's quite cell-specific, and if you know which cell you want to target, that is probably well tolerated.
The chap in the white shirt. Feels like a one-question time.
Have you looked at changes in your cell atlas with age, and particularly in the very old?
Yes, so there are published aging cell atlases. We've also done, our lab has done an aging cell atlas in muscle. The work I'm most familiar with is actually the work we've done on COVID, where we asked, which was a study that I led, where we compared the response of COVID in kids and the elderly. And yes, you see really small differences, because obviously when you're looking at age, we've talked about patient heterogeneity. Often the signals you get are really, you have to work quite hard to dig them out of your data, but I would say that single cell sequencing will find them. And actually the other thing that we found is that when we take cells into culture, they still maintain those age-related phenotypes. So we did alley cultures from children, so airway liquid interface cultures from children and elderly, and infected them with COVID. And actually in the elderly, the COVID multiplied much more productively, and we could show that that was because the cells had a more fibrotic response to injury, whereas the children did not have that. So yes, you can find these things.
Brilliant. Well, thank you again, Kerstin.